Good morning, students. This is Dr. Divya. Here I am teaching you about Nostoc. So this is the taxonomic position. Kingdom Bacteria, phylum Cyanobacteria, class Cyanophyceae, orders Nostocales, family Nostocaceae, and genus Nostoc. Occurrence. So common in freshwater ponds, the large colonies are free floating, appears as circular balls. They may be attached or submerged. It can also be found in soil and moist rock at the bottom of lake and springs, like fresh and salt water, but rarely in marine habitat. So it can also show symbiosis with plant tissues like in hornworts and provide nitrogen to its host through heterocyst. So it may also found as a part of lichen. Plant body. They found in form of colony, cell structures, cell wall, pigments, nucleoplasm, reserved food material. I'll discuss about this. First, colony. So many twisted trichomes arrogate in a gelatinous matrix to form a bowl-like globular colony which ranges from greenish to bluish green in color. And the colony is externally bound by a tough, firm, and pellicle membrane that provides a definite shape to this cell structure. So it is a prokaryotic type, and the cells are without any mitochondria, endoplasmic reticulum, and Golgi apparatus. It has following characteristics: cell wall, pigments, and nucleoplasm. You can say there are single vegetative cells, and in B section B you can see heterocyst. And cell wall, so it's made up of cellulose and it's around the proplast. Pigments. Plastids are absent. The pigments are present inside chromatoplasm, and the cells are blue in color due to the presence of phycocyanin, blue pigment. And other pigment includes chlorophyll, carotene, and phycoerythrin. Nucleoplasm. A definite nucleus is absent, and nuclear matter is present as a central body. It lacks nucleolus. Reserved food material, so it is in the form of sugar, glycogens, and proteinaceous material called cyanophycein. Reproduction, so it reproduces by following methods. First is colony fragmentation, second one is harmogonia formation, third is echinate, and fourth orthospores, and fifth endospores. So it produced by five methods. So first is colony fragmentation. So the colony breaks down into two and more fragments accidentally, and due to some physiological reason, each fragment grows into a new colony. Second one is harmogonia. So it is the most common method in Nostoc. The filament is broken down into a many short length pieces that are called harmogonia, and these type of fragments are formed due to the development of astrocyst or death of any vegetative cell. So these type harmogonia come out of the colony by piercing the colonial sheath. Each develops its own sheath and aggregate to form a new colony. Third is echinate. So these are also called resting spores. A vegetative cell enlarges and secretes a thick wall round and then stores a large amount of food and function as a kinate. They are usually present adjacent to the heterocyst, singly or in the chains, so they help plant during unfavorable conditions. And when conditions become favorable, these echinates release their contents through a pore that germinate into a filament. Orthospores, so the resting echinates are called orthospores. So germination of echinate of orthospores like that. Endospore. So in some species like Nostoc microsporium and Nostoc communium Heterocyst content divide to produce endospores, and these type of spores are released into a new filament. Thank you.